From 2020 to 2022, our planet faced an unimaginable crisis as COVID-19 swept across the globe, forcing millions into isolation, crippling life on Earth and choking healthcare facilities. More than 7 million people worldwide succumbed to the virus. Families were separated, communities were shaken. All of us, teachers, parents, students, had to find a new way to do our respective jobs. As a 7th grade science teacher, I had to quickly adapt, teach atoms, the periodic table, and Newton's laws from my dining table, troubleshoot tech, and keep students engaged and motivated through a screen. We built molecular models on Zoom. We did chemistry with kitchen ingredients. We learned not just science, but resilience. As the world anxiously awaited a way out of the crisis, behind the scenes, a scientist, quiet, determined, and nearly unknown, came to the rescue. Her name, Professor Katalin Karikó. The scientist who made the mRNA COVID vaccines possible, helped save millions of lives during a global pandemic, and never gave up, even when the scientific community gave up on her. This is her story. Childhood. Karikó was born in 1955 in the small Hungarian town called Kisújszalás. Her father was a butcher, her mother a bookkeeper. Karikó grew up in a modest home, which had no refrigerator, no running water, and no television. Surrounded by nature and encouraged by a family that placed a strong emphasis on education and learning, young Karikó developed an early and enduring passion for science. School education. At 13, Karikó traveled nearly 150 kilometers to Budapest from her home for a week-long science competition. She was not accompanied by parents, only a schoolmate. Her father saw her off at the train station and would not hear about the outcome until she returned. When she came home with 3rd place, it was not just a medal. It was proof that Karikó belonged in science. Proof that a girl from a modest home could stand out. In high school, a teacher gave her a copy of The Stress of Life by Hungarian endocrinologist Hans Selye. His message resonated deeply in Karikó's life. Adopting the right attitude can convert a negative stress into a positive one. That idea helped shape how she faced setbacks, not as roadblocks, but as chances to grow and improve. As Karikó would later say, life is like the path of scientific discoveries, never a straight line, with twists and turns at every juncture. Work hard and stay on track. No matter how winding that road may be, it will be the most rewarding journey you never expected. University of Szeged. Karikó pursued her passion for science at the University of Szeged in Hungary, where she earned her PhD in biochemistry. Her research focused on RNA, the molecule that plays a crucial role in translating genetic information into proteins. But before we dive deeper into her groundbreaking work, let us first understand what DNA and RNA are. RNA versus DNA. What is the difference? DNA, or deoxyribonucleic acid, contains all the genetic instructions an organism needs to develop and function. It is found primarily in the cell nucleus and consists of 2 strands twisted into a double helix. The building blocks of DNA are nucleotides, each made up of a sugar, deoxyribose, a phosphate group, and a nitrogenous base. DNA's nitrogenous bases include adenine A, thymine T, cytosine C, and guanine G. RNA or ribonucleic acid is usually single-stranded and plays a key role in carrying out the instructions encoded in DNA. It helps build proteins and regulate gene expression. RNA's nucleotides contain the sugar ribose, the same phosphate group as DNA, and nitrogen bases adenine A, uracil U, cytosine C, and guanine G. Uracil replaces thymine in RNA. Unlike DNA, RNA is more flexible and can move easily within the cell to do its jobs. In summary, DNA stores genetic information, while RNA helps use that information to make proteins. After completing her doctorate, Karikó continued her work at the Biological Research Center of the Hungarian Academy of Sciences in Szeged. However, in 1985, amidst economic challenges in communist Hungary, Karikó's lab lost its funding. On her 30th birthday, she received the news that her position would be eliminated. Determined to continue her scientific pursuits, she sought opportunities abroad. Immigrating to America. Karikó received offers from institutions in London, Madrid, and Montpellier. These European labs were conducting similar RNA research. However, they required her to secure her own funding, a challenging prospect given Hungary's economic constraints and restrictions on transferring money abroad. At the time, Hungary was still under communist rule, behind the Iron Curtain, making immigration and access to Western financial resources particularly difficult. Consequently, Karikó accepted a fully funded postdoctoral position at Temple University in Philadelphia, which provided the necessary resources and support to continue her RNA research. Due to the Cold War era currency export restrictions in Hungary, Karikó and her husband sold their car on the black market and exchanged the proceeds for British pounds. To bypass the restrictions, they hid the money, about 900 pounds or $1,100 at the time, inside their 2-year-old daughter's teddy bear. This was their entire life savings. Arriving in the United States with limited resources and no contacts, Karikó faced the daunting challenge of starting over, driven by a deep commitment to science and freedom. Temple University. Between 1985 and 1988, Karikó served as a postdoctoral researcher in the Department of Biochemistry at Temple University School of Medicine, working under Professor Robert Suhadolnik. During this period, she was involved in pioneering clinical trials that explored the therapeutic potential of double-stranded RNA or dsRNA in treating patients with HIV, hematologic diseases, and chronic fatigue syndrome. What is dsRNA? dsRNA is a molecule made of 2 complementary RNA strands similar in shape to a DNA double helix. It is often found in viruses, so when introduced to the body, it can trigger a strong immune response, especially the production of interferons, which are proteins that help fight off infections and slow cancer cell growth. At the time, scientists did not fully understand how dsRNA triggered the body's immune system to respond, but they knew it stimulated the production of interferons. This ability made dsRNA a very exciting focus for research into treating serious diseases like HIV and blood cancers. Uniformed Services University of the Health Sciences in Bethesda, Maryland, 1988 to 1989. Karikó's departure from Temple University in 1988 was marked by professional and personal challenges. When she accepted a position at Johns Hopkins University, her supervisor, Professor Suhadolnik, reacted negatively by threatening her with deportation and reporting her to U.S. immigration authorities. This led to the withdrawal of the Johns Hopkins offer and difficulties in securing new positions due to Suhadolnik's continued negative references. Eventually, she found a role at the Uniformed Services University of the Health Sciences in Bethesda, Maryland, where she deepened her knowledge of interferons and molecular biology while continuing her RNA research. Despite the new opportunity, she endured a long and exhausting commute between Bethesda and her home in Philadelphia, demonstrating her resilience and unwavering dedication to doing science. University of Pennsylvania. After her one-year tenure at the Uniformed Services University of the Health Sciences, Karikó joined the University of Pennsylvania's School of Medicine as a research assistant professor. She was hired by cardiologist Dr. Elliot Barnathan to collaborate on messenger RNA or mRNA research, focusing on its potential in cardiovascular applications. What is mRNA? mRNA or messenger RNA is a molecule that carries genetic instructions from DNA to the ribosomes in a cell where proteins are made. Think of it as a temporary blueprint that tells a cell which proteins to build and when. Karikó's early experiments demonstrated that mRNA could be introduced into cells to produce specific proteins, laying the groundwork for future therapeutic applications. Despite the promise of her research, Karikó faced significant challenges at the University of Pennsylvania. As a non-tenure-track researcher, her position depended on securing external grants, a task made especially difficult by widespread skepticism about mRNA as a therapeutic tool. Many in the scientific community viewed mRNA as too unstable or likely to trigger harmful immune responses, rendering it impractical for clinical use. Repeated grant rejections eroded the university's confidence in her work, and in 1995 she was demoted from her senior research position to a lower ranking role with reduced compensation. Turning point. A turning point came in 1997 when Karikó met immunologist Dr. Drew Weissman at a campus photocopier. Their shared interest in mRNA led to a groundbreaking collaboration that combined Karikó's expertise in RNA biology with Weissman's knowledge of immunology. One major challenge Karikó and Weissman addressed was mRNA's tendency to provoke strong immune responses, a phenomenon known as immunogenicity. This was partly due to the presence of certain nucleosides in synthetic mRNA that the immune system recognized as foreign. Nucleosides are the building blocks of RNA, consisting of a nitrogenous base attached to a sugar molecule. In natural mRNA, one of these nucleosides is uridine, which is made of the base uracil linked to a ribose sugar. By 2005, Karikó and Weissman discovered that replacing uridine with the naturally occurring variant called pseudouridine could reduce the immune system's reaction. Pseudouridine has the same components as uridine, but differs in the way the uracil base attaches to the sugar, forming a carbon-carbon bond instead of a nitrogen-carbon bond. This modification made the mRNA less inflammatory, more stable, and better tolerated by cells, significantly improving its potential for therapeutic use in vaccines and other treatments. After the groundbreaking 2005 discovery that modifying mRNA nucleosides could reduce its immunogenicity, Karikó and Weissman faced initial skepticism from the scientific community. Their seminal paper was rejected by major journals, eventually being published in the scientific journal Immunity. Despite the lack of immediate recognition, they continued their research, demonstrating that modified mRNA not only reduced immune activation but also enhanced protein production. To deliver fragile mRNA safely into human cells, they used lipid nanoparticles or LNPs, tiny fat-like bubbles that protect the mRNA and help it enter cells efficiently. These LNPs act like delivery trucks, shielding the mRNA from degradation and ensuring it reaches its target inside the body. RNARx. In 2006, they co-founded RNARx to advance RNA therapeutics, securing patents for their innovations. However, they faced persistent funding challenges and limited commercial interest. The University of Pennsylvania sold the patent rights to a private company for a modest sum, leaving Karikó and Weissman without control over their own innovations. During this time, Karikó remained focused on improving mRNA technology, but her position at Penn offered little recognition or opportunity for advancement. Despite the importance of their discoveries, mainstream pharmaceutical companies and academic institutions remained skeptical about mRNA's potential. Karikó's work remained largely behind the scenes, with limited visibility in the broader scientific world. BioNTech. After years of limited support in academia, Karikó left University of Pennsylvania in 2013 to join the German biotechnology company BioNTech as senior vice president. At BioNTech, she found the freedom and resources to advance mRNA-based therapies in a setting that valued her vision. Karikó's work proved critical in 2020 when BioNTech partnered with Pfizer to develop one of the first mRNA COVID-19 vaccines, an achievement that validated decades of research and reshaped the future of vaccine development. Karikó's scientific contributions were also foundational to Moderna's vaccine, making her research the backbone of both groundbreaking efforts to combat the pandemic. She remained at BioNTech until 2022, playing a central role in the rapid advancement of mRNA vaccine technology. How the COVID vaccine works. The COVID-19 vaccine, developed by BioNTech and Pfizer, was built directly on the decades-long research of Professor Karikó and Professor Weissman, especially their groundbreaking 2005 discovery that made mRNA safe and effective for use in the human body. Here's how the COVID vaccine works, step by step. Step 1. Karikó and Weissman's key innovation, modified mRNA. synthetic mRNA naturally provokes a strong immune response, often causing harmful inflammation. Karikó and Weissman discovered that by replacing the nucleoside uridine in the mRNA with pseudo-uridine, the immune system no longer treated it as a threat. This breakthrough reduced inflammation and allowed mRNA to survive long enough to do its job safely inside human cells. Step 2. The vaccine blueprint. Once scientists sequenced the SARS-CoV-2 virus, they identified a critical gene, the one that encodes the spike protein, which the virus uses to invade human cells. That genetic code was synthesized into modified mRNA. Step 3. LNP delivery, the protective bubble. Because mRNA is extremely fragile, it was enclosed in lipid nanoparticles or LNPs, tiny fat-based spheres that protect the mRNA and help it enter cells, like a padded envelope shielding a glass ornament in the mail. Step 4. Inside our cells. After injection, the LNPs fuse with cells and release the modified mRNA. The cell's internal machinery reads the instructions and temporarily produces harmless copies of the virus's spike protein. Step 5. Immune training. The immune system detects the foreign spike protein, triggering the creation of antibodies and memory cells. This training prepares the body to rapidly recognize and destroy the real virus if it ever invades. Thanks to Karikó and Weissman's foundational work, it was possible to develop, test, and deploy the Pfizer BioNTech vaccine in less than a year, a historic breakthrough in science and global public health. Nobel Prize. In 2023, Professor Karikó and Professor Weissman were awarded the Nobel Prize in Physiology or Medicine for their groundbreaking discoveries that enabled the development of mRNA-based vaccines. Karikó's work also earned her several other prestigious honors, including the 2021 Lasker-DeBakey Clinical Medical Research Award, the 2021 Princess of Asturias Award for Technical and Scientific Research, the 2021 Horvitz Prize, the 2022 Breakthrough Prize in Life Sciences, and the 2022 Japan Prize, among many others. In a generous act reflecting her commitment to science and education, she donated a significant portion of her award money to support young scientists and educational initiatives in Hungary. Karikó's discoveries have opened new frontiers in medicine, with mRNA technology now being explored for use against cancer, autoimmune disorders, and emerging infectious diseases. Her perseverance through decades of skepticism and setbacks has transformed modern medicine and continues to inspire future generations of scientists. We are forever grateful to you, Professor Karikó, for saving humanity during the pandemic. Your pioneering research in mRNA technology led to the creation of the first mRNA vaccines, an achievement that not only helped end a global pandemic, but holds the power to revolutionize medicine far beyond the pandemic. Your story teaches us that perseverance prevails, and that holding on to one's purpose and principles, even in the face of rejection, has the power to change the world. Please subscribe, like, and share for more science stories. Happy learning. Thank you.